So today I'll be doing a lesson over the phases of mitosis. The standards that I'm going to be using is develop and use models to communicate the role of mitosis, cellular division, and differentiation in producing and maintaining complex organisms. And the objective is students will demonstrate their understanding of the process by mitosis by completing a virtual online lab, which they will do after I do this presentation. So we're going to do a little bit of vocab review to start off. So Molly, can you tell me what chromosomes are without reading the board?
Let's see. Aren't chromosomes like bundled up DNA?
Yes, you're exactly right. And that bundle of DNA carries genes that code for different things like our hair color and our eye color. How about, Becca, can you tell me about the centrioles?
That's like where the spindles kind of like develop. It's like on either side of the cell.
Good. And then the spindle fibers. Anna, can you just read that off the board for me?
Sure. String-like protein structures that help separate chromosomes.
Perfect. So that's stuff we reviewed and did vocab on yesterday. So there are 5 phases in mitosis. There's interphase, prophase, metaphase, anaphase, and telophase. We'll talk a little bit about interphase first. So in interphase, the DNA is going to be replicated. This is going to make the cell grow a little bit bigger because there's going to be double the DNA that you normally have. And the cell is pretty much going to get ready to separate into 2 different cells. It still has a nuclear envelope, as you can see. There's still a little bit of line. There's that orange dot that is our nucleus. And then it's a diploid cell. Okay, so the next phase is prophase. The chromosomes are going to condense into those little X's that you see on the board. The nuclear envelope is also going to begin to separate and disappear, but it's not completely gone at this phase. It's still there. So when you're looking for a cell that's in prophase, you're going to still see that nuclear envelope. The next phase is metaphase. So in metaphase, the chromosomes line up at the center of the cell. This is pretty easy to determine because if you look at the picture on the board, all of those X's are in the middle of the cell. Also, you'll see the spindle fibers that have formed from the centrioles and are all linked to each chromosome. And as you can see also, there's no nuclear envelope anymore. All right, the 4th phase is anaphase. So in anaphase, the X chromosomes are going to move to each opposite ends of the cell. This one kind of looks like metaphase, but not because the chromosomes are at different sides of the cell. And also, there's still no nuclear envelope. And the last phase is telophase. In telophase, the chromosomes are going to be unraveled. They're not going to be in their X form anymore, and their nuclear envelopes are going to reform. The cytoplasm, though, around the cell, so the cellular membrane has not separated yet. That is a different process that we call cytokinesis. So cytokinesis is not a phase of mitosis. Cytokinesis it's when the cell, like the cytoplasm of the cell, separates completely. So the cellular membrane has separated into 2 different cells. So this is the overall, all the phases of mitosis. As we are doing this lesson, you guys will fill out a handout with these phases each on them, and so that you can refer back to those when we do our little lab. So our in-class activity, I'll hand out another handout that supplies you with the link to this lab, and you'll look at little pictures of each phase of mitosis, and you'll separate them into different groups and count up how many were in each phase, and you'll figure out which phase is the longest and how many, that by determining which one had the most cells in it. And that is it. Thank you for watching.